Imagine a human being, an ordinary woman whose heart stopped beating forever over 70 years ago. Her lungs ceased taking in air, her consciousness dissolved into nothingness, and her body was surrendered to the cold, unforgiving earth, where time and microorganisms did their usual work. But today, right now, at this very second, parts of her body are still alive. And not just alive, they are more viable than you or me. They breathe, they consume food, they reproduce uncontrollably. If we could gather all her living flesh, currently scattered across laboratories worldwide, this mass would weigh more than 50 million metric tons. That is equivalent to the weight of hundreds of skyscrapers made of pure, pulsating human tissue. Her flesh has flown into space. It has been exposed to nuclear radiation. It has been injected with the deadliest viruses known to humanity, and yet it refuses to die. How could an ordinary woman from Virginia become the longest living and most massive organism on our planet? And what terrifying, unnatural mutation turned her cells into an immortal biomass that will outlive us all? To comprehend the scale of this horror, we have to gaze into the microscopic abyss of our own bodies. Every cell inside you has a built-in biological clock. It is a safety mechanism, an invisible executioner, patiently biding its time in the shadows of your DNA. Scientists call this the Hayflick limit. Every time a cell divides to renew your tissues, the protective caps at the ends of its chromosomes, the telomeres, get a little bit shorter. Picture the plastic tips on the ends of your shoelaces. With each cycle of division, this plastic wears away, exposing the threads. When the telomere disappears completely, the DNA begins to unravel. The cell instantly recognizes its own degradation and initiates a self-destruct protocol, apoptosis. It quietly dies, breaking down into safe fragments to protect the entire body from genetic chaos and mutations. This is the natural order of things. It is exactly why our skin ages. It is why our organs wear out. It is why we die. But in 1951, inside the body of 31-year-old Henrietta Lacks, a perfect, catastrophic biological storm occurred. A silent invader, the human papillomavirus, HPV, infiltrated the DNA of her cervical cells. Normally, the immune system obliterates such a threat, or the infected cells simply die off. But Henrietta's cells didn't die. The virus, like a burglar, found a hidden switch within her genetic code and flipped it. It triggered the production of an enzyme called telomerase. In the realm of microbiology, telomerase is a cheat code for immortality. Every time an infected cell divided and its telomere shortened, the enzyme instantly, and with terrifying efficiency, rebuilt it. The biological clock was smashed to pieces. The cells forgot how to die. They became truly immortal. But this eternity came with a terrible price, an insatiable, monstrous hunger for constant growth. Try to visualize what was happening inside her body. A microscopic knot of rebellious flesh, dark and glistening, began drawing blood vessels toward itself like a spider spinning a web. This mass was siphoning off nutrients meant for healthy organs. It was growing not by the year, but by the hour. The doctors who examined Henrietta in the ward at Johns Hopkins Hospital described the tumor as something unimaginable. It looked like smooth, shiny purple jello, incredibly fragile, bleeding at the slightest touch. This was no longer just tissue. It was an aggressive, living entity that had declared war on its own host. When a surgeon sliced off a tiny, dime-sized piece of this glistening flesh and dropped it into a glass tube filled with chicken plasma and bovine embryo extract, something happened that defied all known laws of science at the time. The cells didn't just survive in their cold, sterile glass prison. They began to feast. They divided at such a frantic pace that within 24 hours, the clear nutrient broth turned cloudy, thick with millions of new, starving cells. They crawled up the sides of the glass, expanding, demanding more space, more food. A human death from aggressive cancer is a comprehensible, albeit brutal, tragedy. The real, bone-chilling nightmare began later, when these cells, given the name HeLa, an abbreviation of Henrietta Lacks, left her body and became a global phenomenon. Scientists were ecstatic. Finally, they had human cells that didn't die in petri dishes after just a few weeks. They started shipping them all over the world. Vials of Henrietta's flesh flew on planes, rattled in mail train cars, traveled in the coat pockets of researchers, packed in dry ice. But humanity vastly underestimated what it had unleashed. HeLa cells weren't just resilient, they turned out to be the apex predators of the microscopic world. Picture a sterile, state-of-the-art laboratory somewhere thousands of miles from where Henrietta passed away. Scientists spend years and millions of dollars carefully culturing monkey kidney cells or human lung tissue, desperately trying to find cures for complex diseases. But HeLa is already there. All it takes is one microscopic drop, an invisible speck of moisture that became airborne when a lab technician carelessly opened a vial in the next room. This invisible spore drifts through the ventilation system and softly lands in an open petri dish of healthy liver cells. To the naked eye, nothing has changed, but down there, beneath the microscope lenses, a quiet, ruthless massacre begins. The HeLa cell anchors itself to the bottom. It devours the sugar from the solution, and it divides. 1 becomes 2, 2 become 4. In a matter of days, this incredibly aggressive, rapidly mutating mass starts to overrun the original tissue. HeLa cells grow so densely, they consume resources so greedily that they literally starve the healthy cells, suffocating them, erasing them from existence. By 1966, researchers worldwide realized the horrifying truth. What they had spent years believing were prostate, breast, or kidney tissues, what they had based their PhD dissertations on and tested drugs against, all of it had been devoured and replaced by HeLa cells. Henrietta's immortal flesh had contaminated and hijacked countless experiments. It was an invisible, unstoppable pandemic of immortal human biomass. If we zoom in even further, down to the scale of a single micrometer, and observe a HeLa cell dividing, we won't see the beautiful, symmetrical dance of nature described in high school textbooks. We will see a chaotic, grotesque distortion of life itself. A normal human cell has 46 neat, highly organized chromosomes. Inside HeLa, there is pure genetic madness. 76, 80, sometimes well over 100 chromosomes. They are torn apart, stitched back together, mutated beyond recognition. When this cell prepares to split in two, it doesn't just pinch softly in the middle, it goes into convulsions. Its membrane violently boils and blisters, shooting out long, spider-like tentacles, Philopodia. These tentacles lash out at the pink nutrient broth, gripping the glass with incredible force. The cell literally rips itself apart with brutal, mechanical power, while its mutated chromosomes tear and reassemble in new configurations. This is life reduced to its most primal, terrifying imperative: reproduce at any cost. This flesh no longer cares about form, function, or harmony. It has become a perfect biological machine, optimized solely for endless, aggressive expansion. Some evolutionary biologists have even put forward a shocking hypothesis. HeLa cells can no longer be considered human. They have drifted so far from our genetic code. They have adapted so perfectly to life in glass jars and steel incubators that they should be classified as an entirely new, single-celled species. They even coined a name for it, Helacyton gartleri, a new form of life, born from the agony of a woman that is now quietly colonizing laboratories on every continent of Earth. Henrietta Lacks died in the cold September of 1951. Her body was utterly consumed by the very same mechanism that granted her biological immortality. The tumors metastasized into her lungs, liver, and intestines, until her organs simply shut down, unable to withstand the monstrous pressure of this biology gone rogue. She was buried in an unmarked grave under a drizzling rain, and for decades, the world forgot her name. But this world had already been forever changed by her. While her bones were turning to dust, thousands of massive vats at factories across America were churning with tons of her living flesh. It was on HeLa cells that the world's first polio vaccine was tested, saving millions of children from paralysis. Henrietta's flesh was packed into capsules and sent into Earth's orbit on the first Soviet Sputniks and American space shuttles to see how zero gravity and cosmic radiation kill living tissue. Without her, we would never have mapped the human genome. We wouldn't have created treatments for HIV and leukemia, and we wouldn't have learned how to clone DNA. We owe an unpayable debt to this tissue. But as you sit here, listening to this voice, feeling your own mortal heart beating, feeling the blood racing through your veins, think about the quiet, dark incubators. Right now, in this very millisecond, in tens of thousands of labs from Tokyo to New York, compressors are steadily humming. Inside, in absolute pitch black, floating in a warm red liquid, Henrietta's flesh continues to divide. It doesn't know what it has accomplished. It has no brain to comprehend its triumph, no eyes to see the stars it has flown to, no soul to find peace. It only knows hunger, and it only knows time. And centuries from now, when you and I are long gone, when our bodies are nothing but dust carried by the wind over a cooling planet, the cells of Henrietta Lacks will still be there, waiting in the cold glass, dividing in the dark, a silent, eternal monument to human fragility, built from the terrifying, undying power of our own flesh. The ocean of time will wash away our names, erase our cities and civilizations, but this flesh will just keep going.